There is a moment most gardeners know, but almost none talk about. You are standing in the garden center aisle holding a bottle. The label is impressive. Words like advanced biostimulant and professional-grade root enhancer printed over a photo of the most perfect tomato you have ever seen. The price tag says $40. And you have been here before. You bought it last summer, the summer before that, and the plant grew. Mostly. The harvest came. Mostly. But there was always that nagging feeling that $40 should have done more. Here's what the garden center never mentioned, and what a single peer-reviewed study published in the journal Crop Science documented back in 2014. A kitchen ingredient that costs less than $1, one most people throw away without a second thought every single week, produces a root mass 2 to 3 times denser than the same variety grown without it by the end of a single growing season. Not 2 to 3% better, 2 to 3 times. The kind of difference you can hold in your hands when you pull the plant at the end of August, when the season is done and the underground truth finally comes to the surface. That ingredient is not at the garden center. It never will be. There is no profit in it. It has no brand name. It cannot be bottled with a logo. And that is exactly why most gardeners over the age of 50 have spent decades walking past it in the kitchen without ever knowing what it could do 12 inches under the dirt. This is the story of what that ingredient is, what the science actually shows, how to use it correctly so the results show up in the real garden instead of just in a laboratory, and the 5 quiet mistakes that prevent most home attempts from working at all. But before getting to the recipe, there is something worth understanding about the underground world most gardening advice never explains. For most of a gardener's life, the eye is trained upward, up at the leaves checking for yellowing, up at the blossoms counting the flowers, up at the fruit measuring progress by what is hanging on the vine. The instinct when something goes wrong is always to look at the surface. The plant looks tired, spray the leaves. The fruit is small, scatter something on the soil. The transplant wilts. Give it more water and hope for the best. But the truth that takes a lifetime in the garden to fully understand is that everything a plant becomes is decided in the dark. Down below the soil line, somewhere between 6 and 18 inches under the surface, is where the entire outcome of a growing season is written before the first blossom ever opens. If the roots are shallow, no amount of liquid fertilizer poured over the top will fix the harvest. If the roots are thin and stringy, the plant will collapse under the first August heat wave, no matter how carefully the watering schedule is kept. If the roots are starved of oxygen, microbial partners, or the right hormonal signals, the leaves above will yellow, regardless of what gets sprayed on them. This is the quiet rule the garden center aisle is built to ignore. Because what sells in a garden center is visible. Leaf sprays with bright-colored caps, bloom boosters in foil pouches, root stimulants in small, expensive tubs. All of it aimed at the part of the plant people can see. None of it aimed at where the real work happens, the engine of the plant. The only structure that ultimately decides whether a pepper swells to full size or a tomato hangs heavy on the vine lives entirely below the soil line. And the question worth asking is, what does a root actually need in order to triple in density? Not what does the label say it needs. What does the biology say? Walk through the rooting hormone aisle of any garden store, and the story told by the labels is almost always the same. Synthetic auxins, indole 3-butyric acid, usually abbreviated as IBA. It is the industry standard, and it works. It prompts cell division at the root tip. Cuttings dipped in IBA powder root faster than untreated cuttings. That part of the science is real. But here is what the label does not say. IBA is a synthetic copy of a hormone that already exists in nature. That hormone is produced naturally by an organism so common it is sold at every grocery store in the country for under $1 a packet. Saccharomyces cerevisiae, baker's yeast, the same powder generations of grandmothers have used to make bread rise. In 2014, a team of researchers published findings in the journal Crop Science showing that when living yeast cells were introduced to the root zone of tomato plants, root biomass increased significantly. In 2023, a study in the journal Plants documented that oxytocin-producing yeast strains used as soil inoculants produced a 1.5-fold increase in plant height, fresh weight, and root length compared to untreated control plants. And in 2024, a paper in Frontiers in Industrial Microbiology confirmed that residual brewer's yeast improved seed germination, leaf number, fresh weight, and chlorophyll content in both lettuce and tomato seedlings. 3 independent studies across a decade, all pointing at the same pantry shelf. Here is how it works, because understanding the mechanism is what allows the home gardener to avoid the 5 mistakes that ruin the results every time. When living yeast cells are introduced to moist soil at the root zone, 3 things happen simultaneously. First, the yeast produces a compound called indole 3-acetic acid, the natural version of the same hormone class the synthetic rooting powders are trying to replicate. IAA, as it is abbreviated, is the master signaling molecule of the plant kingdom. It is the chemical message that tells a root cell to divide, to stretch, and to push out a new root tip. The plant does not need to manufacture it. The yeast delivers it directly. Second, as the yeast cells multiply in warm water, they release amino acids, peptides, B vitamins, and trace minerals including nitrogen, phosphorus, potassium, magnesium, iron, and zinc directly into the surrounding liquid. These are not locked up raw minerals that require weeks of microbial breakdown before the plant can access them. They are already in the pre-digested, immediately absorbable forms that root hairs drink on contact. The closest thing in nature to a direct intravenous line for a stressed plant. Third, the yeast cells settle into the root zone and physically occupy space. They compete with soil-borne pathogens for nutrients and for territory. Studies on lettuce and tomato have noted lower incidence of certain damping-off diseases in yeast-treated root zones, attributed partly to that biological competition and partly to antimicrobial peptides the yeast secretes while it grows. 3 effects from 1 teaspoon of pantry yeast: hormone signal, nutrient delivery, microbial defense. And there is one more piece of this story, the piece that surprised plant scientists when it was first documented under a microscope in 2010. For most of agricultural history, roots were considered passive. Straws sipping dissolved minerals from damp earth. That was the textbook picture for over a century. Then, researchers at the University of Queensland used confocal laser scanning microscopy, a kind of fluorescent imaging that watches living root cells in real time, to observe what actually happens when living yeast cells meet a young tomato root. What they saw changed the model. The root did not just absorb nutrients passively, the root actively changed its growth direction to move toward the yeast cells. It branched toward the signal source, sending new lateral roots out like fingers, reaching for something they already recognized. Yeast had been growing alongside plant roots for tens of millions of years before the first human ever walked into a garden center. The root system was already built to use it. Now, the recipe. Because this is where most videos on this topic lose credibility by making it sound either too complicated or too vague. There are 3 ingredients, and the preparation takes 30 minutes. 1 liter of non-chlorinated water, 2 tablespoons of plain dry baker's yeast, roughly 20 grams or 2 standard packets, 1 teaspoon of plain white sugar. That is the complete ingredient list. The total cost, depending on the yeast brand, is somewhere between 60 cents and $1. Step 1 is the temperature, and this is the step where most home attempts quietly fail before they even start. The water must be lukewarm. The accepted activation range for dry yeast is between 105 and 115 degrees Fahrenheit. Both Bob's Red Mill and Fleischmann's published the same window. The simplest test for anyone without a thermometer is to touch the side of the jar with the back of the wrist. The water should feel comfortably warm, slightly more than body temperature, not hot enough to pull the hand away. Water hotter than 120 degrees kills the yeast cells before they activate. The brew will smell sour rather than bread-like. If that happens, pour it out, use cool, fresh water, and start over. Step 2. Add the sugar and stir until dissolved. The sugar is food. It is what the yeast cells consume first, what triggers the initial bloom of activity. Without it, activation is sluggish and unreliable. Step 3. Add the yeast. Stir briefly and cover the jar loosely. A paper towel secured with a rubber band is the ideal cover. This is the most common mistake beyond temperature. Sealing the jar with a tight lid. As the yeast activates, carbon dioxide pressure builds quickly. A glass jar sealed tightly on a kitchen counter can crack without warning. A paper towel cover is the complete and only solution needed. Within 40 minutes, the surface of the liquid should show bubbles. Within 2 hours, a thick foam will have formed at the top. That foam is the yeast colony at full activity. This is the concentrate. It is not yet ready to touch the garden. Step 4. Dilution. Take 1 part of this active concentrate and mix it with 9 parts of plain water to make the application solution. Do not skip this step. Undiluted yeast concentrate applied directly to a root zone can cause mild root stress from the concentrated organic acids. Properly diluted, it is gentle enough to use every 2 weeks through the entire growing season. Step 5. Timing of application. Pour the diluted solution around the base of the plant in the early morning or in the last hour before sunset. Never in the heat of midday, when soil temperatures are at their peak. The yeast population can be stressed by root zone temperatures above 95 degrees Fahrenheit. Cool soil absorbs and holds the solution better, and the plant drinks through the night at its own pace. The solution does not keep. Use it within 24 hours of mixing. After that, the yeast population crashes and fermentation shifts toward alcohol, which can mildly stress root tissue rather than support it. The right timing across the season matters just as much as the recipe itself. There are 3 application windows where yeast water produces measurable results, and applying it outside of these windows wastes the brew and misses the opportunity. The first window is at transplant. When a seedling moves from a tray or a small pot into the open garden, it enters a period of stress that plant scientists call transplant shock. The roots have been severed, bent, or compressed. The microbial partnerships the plant spent weeks building in the original growing medium have been disrupted. A single application of diluted yeast water poured around the root zone at the moment of transplanting gives the new root tips a direct supply of IAA and immediately begins establishing a new microbial community in the surrounding soil. Plants treated at transplant hold their leaves more firmly through afternoon heat within the first week. New lateral roots establish faster. Main stems visibly thicken within 2 to 3 weeks. The second window is at the onset of flowering. The plant has shifted from vegetative growth to reproductive mode. Its demand for phosphorus and trace minerals spikes sharply at this moment, because those are the raw materials for building flower tissue and setting fruit. A second yeast application at the first sign of flower buds delivers another wave of readily available trace minerals and continues the hormonal signal to the root system to keep branching and deepening. The third window is when the first small fruits appear. The plant now needs to pull in heavy nutrient loads to swell each fruit to full size. A third wave of yeast-supplied amino acids and minerals at this stage supports the demand without forcing the plant to compete with itself for resources it does not have enough of. 3 applications spaced across the season. That is the entire system. More frequent applications risk shifting the soil microbial balance too far in one direction. Fewer than 3 misses the windows when it would have made the measurable difference. Now for the honest part, because this is where most gardening videos quietly lose the trust they spent 10 minutes building. A single application of yeast water will not transform a struggling plant overnight. The visible changes arrive gradually. The most dramatic evidence of what the yeast actually accomplished shows up not while the plant is growing, but at the very end of the season, when the tomatoes are done and the plant gets pulled from the ground. Pull a yeast-fed tomato plant at the end of August, and the root ball will not look like it belongs to the same plant as an untreated variety grown beside it. The yeast-fed root mass is often 2 to 3 times denser, visibly more fibrous, with significantly more lateral branching radiating outward through the soil. That is the experience behind the title of this video. The $40 bottle promises results you can see on the label. The $1 packet of baker's yeast delivers results you can hold in your hands at the end of the season, when the truth of the soil comes up with the roots. The plants that respond most strongly are the heavy feeders. Tomatoes, peppers, eggplant, cucumber, zucchini, and corn show the clearest gains. Strawberries respond well, particularly during the runner stage. Leafy greens, including lettuce, spinach, kale, and chard, show measurable improvement during the seedling and transplant phase specifically. Root crops like carrots and beets benefit primarily from the early establishment window and respond less dramatically to later applications. There is something quietly satisfying about a garden recipe that begins in the back of a pantry drawer, rather than in a brightly lit store aisle. The same yeast that has made bread rise in farm kitchens for generations also makes a root system reach deeper and branch wider. The earth has been generous with its tools all along. The most useful one was sitting on the kitchen shelf the whole time. If this kind of practical, evidence-backed gardening wisdom is what brought you to Rooted Grace, the like button and the subscribe button keep this work going. There is one more homemade preparation that pairs naturally with yeast water and works through a completely different mechanism in the soil. A recipe that, used at the right time, can double flower count and change the color and density of the harvest. That one is coming up next. Until then, may every plant in your garden hold deep, and may the harvest be heavy.